So this is the final slide then of the manufacturing process for Januvia, for the sitagliptin, the active ingredient in Januvia. We take the enamine amide, we then subject it to the asymmetric hydrogenation conditions, as I'd mentioned. Here it's only 95% EE. To get a drug approved in the U.S. and for most other countries, you have to have greater than 98% enantiomeric excess because you cannot have more than 1% impurity of a compound at that type. So we didn't have to do a crystallization. We can do that very easily. The other thing about this process, rhodium is a very expensive metal, and we want to be able to recover the rhodium. And we can do that very simply by taking activated charcoal. Ecabsorb is the commercial name. It's just activated charcoal. You throw that into the reaction mixture. The rhodium binds to the activated charcoal. You can simply filter that, and thereby we can recover 95% of the rhodium by just taking that charcoal, incinerating it, capturing the rhodium back, and we can reuse the rhodium. And we can do that with our colleagues in the heavy metals business. So it's a very green process from that perspective of reusing the very precious transition metal. Then after we do that, we do the final upgrade by doing a crystallization in isopropyl alcohol and heptane. And then that material is subjected to the conditions of water with phosphoric acid, which then generates the active ingredient, the phosphate salt of sitagliptin. This process, as you can see on the bottom, we've made metric tons of sitagliptin, and this is how we initiated the chemistry for the New Drug Administration, our application rather, which occurred in 2005. Just a thing of note that Chris highlighted and others have highlighted from the panel discussion this morning, one of the driving problems in pharmaceutical research is the time cycle it takes from the discovery of a drug to the development of the process to go through the phase 1-3 clinical studies, and then you go for an NDA. The average is 12 to 15 years in industry. We were very fortunate with this molecule, however, that we did the whole process in 3 years and 10 months. And I'll say that again, 3 years and 10 months. So what occurred was, again, we used partnerships to bring that to fruition, right? The first thing that we did, we looked at the novel field of DPP-4 inhibitors, okay? There was a small company in the Netherlands that was pursuing this called ProBio. So we did an agreement with them, we brought their molecules in, and we found very quickly those molecules had tremendous safety issues with them. And so we then set up a huge screen in our biological laboratories to understand the toxicity from those first molecules that were DPP-4 inhibitors. It turned out they had DPP-4 activation and DPP-8 activation. And the DPP-8, the other enzyme, was the one that gave the toxicity. So by dialing that out with our discovery chemists in relationship with the biologists, and they did that in less than a year, we then discovered the molecule Januvia. When that compound then came into development, as Chris highlighted in his talk, we were thinking quite some time about how could we make these beta amino acids, and we had to make that moiety to support the campaigns. Well, we got very, very lucky. Also, there's serendipity of why you have a process that takes 3 years and 10 months versus the 12 years. We got lucky. You know, for the first campaigns, our yields doubled, and then we were able to quadruple those yields based on some tweaks that we did. And then when we partnered with Solvius from Switzerland, we leapfrogged what we could do with this chemistry. And at the time, that type of chemistry was unprecedented at scale. No one had ever done that kind of chemistry for the last step, because you're introducing a heavy metal in the last step, and that's usually taboo. You don't want to do that because you can't have a heavy metal residual in a pharmaceutical product. But we had great confidence that we could remove that and purify that material so we could pursue that type of chemistry. The next thing I want to switch gears on, and just before I do that, this shows you where we started with the first pilot plant campaign that we did for the molecule for the first 100 kilos and what the manufacturing route is today. So you can see this is the amount of waste now generated in its entirety versus the blue bars show the waste that we had for the initial campaign. So again, it really shows how innovation can obviate waste and really allow you to have an environmentally benign chemistry that's sustainable. And we were very fortunate to not only win the European Prize for Green Chemistry, but also the Presidential Prize for Green Chemistry based in the U.S. So I want to switch gears a bit. So we as a company with Merck, we always are looking for where's the next level of innovation we can bring. And as I mentioned, we actually tried to pursue a biocatalytic version of taking a ketone to that chiral amine 10 years previously. but we were completely unsuccessful. And we actually partnered at the time with a company to pursue this chemistry, but they did not have the technology in place that a company like Codexis has. The aspirational way to make this molecule is shown on the next highlight. What you would love to do, as I highlighted, is to take this ketone, transfer that with an amine moiety to give you this molecule directly with the chiral amine. And so a biocatalytic process. It should even be a greener process, but the bar is very high because we have a very efficient process. So how can we actually come about to bring that together? And the only way that we could do that is with a company called Codexis. They have embedded technology of enzyme evolution, where they change the structure of the enzyme through genetic evolution. That then allowed them and allows us as a team to develop this chemistry. And let me just highlight that for you very quickly. So what's known is that there are transaminases that exist. That's a type of enzyme that does the reaction that's highlighted on this slide, where you have an amine acceptor and an amine donor, and they kind of just switch places, where the amine goes there and then you generate the ketone. And these are very ubiquitous in nature. They're quite common, actually. But there's a few drawbacks. Typically, the way that this chemistry works is quite complicated. This just shows the catalytic cycle of how this chemistry works. So I'm not going to go through it. I just want to highlight the complexities of this chemistry. And more importantly, the challenging part, as this thing highlights, it always wants to exist as this molecule, the ketone, not as the chiral amine. So you have to trick nature. You got to trick it somehow so that you get the molecule that you want. Or you have to do some other type of technique, and I'll describe that just very quickly. So this is the classic approach that people had in the past for doing biocatalysis for reductive amidase. They use a quite sophisticated amine source, and then to manage that, you would have to reduce the byproduct to something else, and you kept doing all these iterations, as you can see here, to try to allow this equilibrium to go from the ketone and stay as the amine. Some of them were somewhat successful, but they were really prohibitive from a cost-wise and from a complexity point of view. There was no way you're really going to scale this to the level that we needed for a molecule of like sitagliptin. So what was the simple solution? It was very trivial. You change the amine source. So if you go back here, this was the amine source that everyone had used for generations in academia. And then in Merck Research Laboratories, we just postulated, what if you use isopropylamine? It goes to acetone. And guess what? Acetone is highly volatile. Just by passing nitrogen over the reaction itself, it will remove the acetone. And based on a classic chemistry principle, if you remove this material, all the equilibrium will flow to the right to the desired amine. So a very simple solution for an extremely challenging problem. And I think that's one of the things that a process chemist can bring to bear in opportunities as this, is that simplicity equates elegance. The simpler you can do something, the more elegant it is, and therefore more environmentally benign and more sustainable. So one of the problems with this approach, though, the type of enzymes that did this transformation could not do a molecule like sitagliptin. They could not do it. And what we found is you had a small group and a large group. And all the molecules that had been shown to use reductive amidase, that was the type of molecule you had to have. But unfortunately, the commercially available transaminases showed no activity for our molecule. So what could we do different? So we looked at the literature and we saw that these molecules had something in common. You could get the R stereochemistry, the desired stereochemistry, and you had a large group and a small group. And then we wanted the chemistry to work on this molecule. And when we tried this molecule with different types of enzymes, absolutely no reaction. So we then did a very simple approach. We said, okay, well, let's just truncate the molecule. Let's just test enzymes initially with with this moiety, with the triazole portion, and then we'll add this portion later. And so then we could trick the enzyme, and we could evolve the enzyme, again, with our collaborator Codexis, because they have this genetic evolution technique where they can actually modify the active site of an enzyme, and they can open it gradually, so then that we first get an initial hit here, we can then evolve that enzyme so it could work with a molecule of that type. This just shows the way it went about and some of the highlight of the results. So we found a very simple transaminase that could do the transformation. We did multiple rounds of iterations, and you can see how the conversion went higher and higher. So after about a year working with Codexis, we were able to get this chemistry when it was initially 0% reactivity, and now we're at greater than 99.9% EE and 95% conversion. And this slide is supposed to be, daunting, because this is actually what Codexis did. So what you need to take home from this slide, so this is the structure of the enzyme, okay? This is the molecule of the enamine amide that's being hydrogenated, all right? So what you see first is they evolved genetically the active site, and that's what's in green. And they did that to accommodate that larger size of the left-hand part of the molecule, as I highlighted. Next, what they did, and their technology and intellectual property, they can then change the enzyme in different places to allow you to use organic solvents instead of water, to allow you to use heat, so you can use vigorous stirring. So you give structure and integrity to the enzyme that nature does not provide. So you evolve the enzyme for it to do what you want it to do. And as I said, you kind of trick the enzyme into actually carrying out the reaction. So that was the technology that Codexis had. And this just shows the final reaction that's being used today. We'll be transferring to this process this year, in 2011 hopefully. And so we take a simple enzyme, use isopropyl ethylamine. We then do the bioconversion. The first issue that we had, this compound kept oiling out in the reaction conditions. We had pH control issues and etc. However, we were able to solve those very quickly by using a little bit of DMSO. We could then add the ketone as a solution over time, which is a very easy trick that we use in process chemistry. We could control the pH with the propyl molarity of the amine. And then with a simple acetone removal, as I mentioned before, with just nitrogen passing over the reactor, it elevates and allows the removal of acetone very simply. So it drives at equilibrium to give us the chirolamine. This process now gave the same purity profile as the original asymmetric hydrogenation process. The other thing that's critical when you change chemistries for a molecule that's already being on the market, then you have to make sure the impurity profile is the same. So this just highlights the chemistry for Januvia for the biocatalytic conversion. And the other thing to mention, this chemistry now last year was also granted the Presidential Green Challenge Award, and it's the only time that a molecule has gotten the award twice. So for the first chemistry that we did, the asymmetric hydrogenation, and then the biocatalytic reaction, it really shows that what we try to do at Merck and what other pharmaceutical companies are trying to do is strive with innovation to evolve the processes that you have, because you can make them greener and better and more cost effective and more sustainable. And it's really about that innovation that drives it. So in conclusion, these are the highlights. What I've tried to show you today are two specific ways that you could actually make the molecule, the active ingredient of Januvia, sitagliptin, from an asymmetric hydrogenation in a biocatalytic way. Also, I've tried to give you a sense that, green chemistry goes hand in hand with good science. It really, at the end of the day, is about the science innovation that you can drive to bring these type of opportunities to fruition. And then lastly, one of the things that we desperately believe in and that we absolutely hold dear at Merck and Company is that our approach to chemistry with innovation really mimics our approach to creating medicines to support the world's population, and that we have to have a green footprint wherever we make those medicines, wherever we give those medicines, and how we discover and develop them and manufacture them. And with that, I'll thank you for your time and consideration.